Genus Eimeria coccidia. Genus Eimeria coccidia. Genus Eimeria coccidia. Phylum Epicomplexa. Class Sporozoa. Members Member of Sporozoa class are parasitic and spore forming. Phylum Epicomplexa has apical complex, polar ring, conoid, micronemes, rhoptrians, micropores, and subpellicular tubules. Both asexual zygogony and sexual gametogony type of reproduction present. Member produce spore by means of sporogony. Phylum Epicomplexa, class Sporozoa. Subclass Coccidia. Order Eucoccidiada. Family Eimeridae, Cryptosporidae, and Sarcosistidae. Family Eimeridae, genus Eimeria. Organism commonly called Coccidia, intracellular parasite found in the epithelial cell of intestine of vertebrae in which both asexual and sexual reproduction take place. Highly host-specific as well as organs or location-specific parasite. Direct life cycle, locomotion by gliding. Family Eimeria, genus Eimeria. Unsporulated oocysts come out of the infected host through feces. Sporulation of unsporulated oocysts occurs outside the host in environment. Sporulated oocyst is the infective stage of the host. Unsporulated oocyst usually spherical, subspherical, ellipsoidal, and oval shape. Usually bilayered, transparent, and clearly visible. A small pore called micropyle may be present at narrower end, which is often covered by a polar cap or micropylar cap. A nucleated mass sporon is founded mostly at the center. Unporulated oocysts come out of the infected host through feces. Sporulation of unsporulated oocysts occurs outside the host in environment. Sporulated oocyst. Sporogony occurs outside the host. Sporulated oocysts are the infective stage of the parasite. Time taken of sporulation can also help as an aid to identification of species. Sporulated oocysts contain different number of banana shape sporozoid, sporocyst, residuum, and stadia body. Each sporulated oocyst contains 4 sporocysts and each sporocyst contains 2 sporozoids. Eimeria species found in poultry. Species site in host disease. Eimeria tenella, cecal coccidiosis. Eimeria necrotrix, small intestinal coccidiosis. Eimeria brunetti, rectal coccidiosis. Eimeria acervelina, small intestinal coccidiosis. Maxima, small intestinal coccidiosis. Mites, intestinal coccidiosis. Pericoque, small intestinal coccidiosis. Eimeria species found in cattle and buffalo. Species and remark. Eimeria zurani, winter coccidiosis most common and pathogenic. Eimeria bovis most common. Eimeria berylli found in water buffalo. Alabamensis E. buccidenososis, biggest species among Eimeria species of cattle and buffalo. Coccidiosis canadensis, Eimeria species found in sheep. Species remark. E. ovina, most common and pathogenic. Intricata, E. intricata, largest coccidiosis species of sheep. E. granulosa, egg or urn-shaped oocyst. Astrata, dome-shaped micropylar cap, Crendalis geruthi, found in abomasum of sheep and goat. Imeria species found in goat. Species and remark E. Arloingi, most common. Caprina, Hirchi, Naina, Kohayakimovi, Gilruthi, found in abomasum of sheep and goat. Imeria species. Species final host site. Species final host site in host. E. Debliaki, pig do. Leucarati, equine, Sam. Truncatata, geese, kidney. E. Citidae, Leeuwenhoek observed first parasite protozoa. Example, Imeria citidae from the gallbladder of rabbit. Rabbit liver. Coccidian species of dog and cat. Species final host. Isospora canis, dog. Isospora oensis, Sam, dog. Neospora caninum, dog. Sarcocystic cruzi, dog. Isospora fellis, cat. Isospora revolata, cat. Toxoplasma gondii, dog and cat. Genus Imeria. Genus Imeria location. An intracellular parasite found in the epithelial cell of the intestine. Infective stage sporulated oocyst. Transmission ingestion of sporulated oocyst along with food or water. Life cycle of Imeria. Life cycle inside the host. Host get infection by the ingestion of sporulated oocyst through feed and water. 3 phase occurs inside the host. Excitation, cyst wall rupture and release sporocyte enter in the alimentary canal in the presence of CO2, bile and trypsin. Sporozoid penetrate the intestinal epithelial cell and form rounded trophozoid. Genus Imeria. Schizogony trophozoid multiply by means of schizogony multiply fission and form schizone containing numerous elongated merozoites. Schizone rupture and release merozoites invade neighboring cell and continue second generation schizogony. Syngamy, merozoid from male microgametocyte and female gametocyte, microgametocyte and macrogametocyte from microgametes and macrogametes respectively. Flagellated microgamete motile are released from the rupture host cell and fertilize the macrogametes. After fertilization, zygote is formed which transformed into unsporulated oocyst and passed out through the feces of the infected host. Outside the host in the environment. Unsporulated oocyst become sporulated outside the host in environment in the presence of O2, suitable temperature and moisture. At the temperature of 20-30 degrees Celsius with sufficient air and moisture, the sporulation occurs within 1-3 days. Life cycle of Imeria. Coccidia life cycle. Subclinical small intestine. Merozoid infect large intestine. Day 16. Clinical large intestine. Day 21. Excretion of oocyst. Day 28. Day 1st. Daily contamination by oocyst. Day 5. Merozoid infect small intestine. Subclinical small intestine. Imeria life cycle. Imeria life cycle. Day 0. Oocyst. Excitation. Oocyst release sporocyst. Sporocyst release sporozoid. Sporozoid invade gut cell. Day 3. Trophozoid. Schizone. Merozoid release from schizone. Male and female gamete develop oocyst. Oocyst. Day 7. Unsporulated oocyst. Sporulated oocyst. Coccidiosis is regarded and coccidiosis is regarded ubiquitous in poultry management. It is considered as a man-made disease in overcrowding and unhygienic conditions encourages young animals or birds are more susceptible and adults act as carriers. Three types of coccidiosis occur in poultry: cecal coccidiosis, rectal coccidiosis, intestinal coccidiosis. Cecal coccidiosis caused by Eimeria tenella. It occurs mostly in young birds of 3-7 weeks of age. Facial hemorrhages found in enlarged cecum on 4 days after infection. Lumen of ceca filled with consolidated and dark brown to blackish caseous mass. Cecal core containing feces, necrosed cellular debris, and blood. Hemorrhages in ceca. Hemorrhages in cecum. Cecal core is characteristic lesions of cecal coccidiosis. Symptoms: dropping, stop feeding, but may continue to drinking water, bloody diarrhea, red diarrhea, anemia, and death. Rectal coccidiosis caused by Eimeria brunetti. Affected bird showed white fluid dropping mixed with blood and mucus, cast, severe dehydration, reduced body weight, and lack of appetite. Intestinal coccidiosis caused by E. necatrix, E. acerulina, E. maxima, and E. maiveti. Generally, older birds are affected and chronic form is common. In E. necatrix, submucosal hemorrhage is found mainly in the middle part of the small intestine. Intestinal coccidiosis: the small intestine is markedly swollen, hemorrhages, and is filled with clotted or unclotted blood. Pinpoint to pinhead size grayish white spots are found in small intestine. Symptoms: watery diarrhea with blood E. nectarix, chronic watery diarrhea without blood E. acerulina. Genus Eimeria. Clinical sign of coccidiosis: slow growth of young birds, felanes, change in dropping diarrhea blood. Coccidiosis in calf. Coccidiosis in calf. Winter coccidiosis caused by Eimeria zorani occurs in winter season. Symptoms: foul smelling diarrhea with or without blood, abdominal pain, soiled hen water, anorexia, unable to stand up, etc. Infected young calves died due to heavy loss of blood. Eimeria diagnosis on the basis of symptoms, bloody diarrhea, etc. Microscopic examination of feces revealed double-walled unsporulated oocyst with nucleated mass. PM finding: diagnosis of coccidiosis in chickens is best accomplished by post-mortem examination of a few affected birds because of major pathogenic effect usually occurs prior to oocyst head in feces. Eimeria diagnosis: 2.5% potassium dichromate solution are used for sporulation of unsporulated oocyst in lab. K2CR27 potassium dichromate solution prevent the bacteria from destroying the oocyst and also provide oxygen to the oocyst for their development. Eimeria species treatment: a number of drugs available which are used either sign or in combination along with feed or water. Sulfadiamine 120 250 ppm, amprolium 6 to 125 ppm, diclazuril 1 to 5 ppm, toltazuril 25 ppm, semiduramycin are other anticoccidial drugs. Linophorus antibiotics, example monensin 100 to 120 ppm, lesalocid 100 to 120 ppm, and salinomycin 50 to 70 ppm. Eimeria species treatment. Amprolium prevents coccidia from utilizing thiamine by blocking thiamine receptors and effective against first generation trophozoite and schizoid. Coccidiosis in poultry is man-made disease and self-limiting disease. Shuttle and rotation program adopt in use of anticoccidial drug in poultry to prevent drug resistance. Primary species control by chemotherapy. Anticoccidial drugs are used as a chemotherapy and chemoprophylactic to control infection. By adopting good managemental practices like litter should kept dry, feeding and water throw should be kept such a high that they cannot be contaminated by droppings. Primary species control. Good ventilation inside to reduce moisture and sporulation of cysts. Overcrowding should be avoided. Clean feed and water should be provided. High proteinaceous feed along with vitamin A, vitamin K, etc. are beneficial for treating and controlling coccidia infection. Primary species by vaccination. Coccivax contain 8 primary species: Immucox nobilis, Paracox 5, Coxsackie, and Levacox e tenella. Coxsackie is the first development subunit. Vaccine comprises a purified protein isolated from gametocyte of E. maxima by a big Israel. Vaccination. Coccivax, Immunocox nobilis are live unattenuated vaccines. Levacox and Paracox are live attenuated vaccines. Attenuation is done by selection for precocious. Thank you.